Hi everyone, it's me Anita Paneru. I am a PhD candidate at Dr. Marine Friesen's lab at Department of Plant Pathology at Washington State University. Today I'm going to talk about engineering soil-derived synthetic microbial communities syncoms for biological control of plant disease. Yeah, welcome to my talk. Let's take a moment to appreciate why soil microbes are important for agriculture and ecosystem. They involve in plenty of functions like nutrient cycling. They break down dead plants and animal material, releasing carbon, nitrogen, phosphorus, and other elements back into forms that plant can readily absorb. So, building on that, certain bacteria perform nitrogen fixation, converting inert atmospheric nitrogen gas into ammonia. So, without this fertilizer, most plant couldn't access enough nitrogen to grow vigorously. At the same time, other microbes mobilize locked up minerals like phosphorus and potassium from soil particle, making them bioavailable to plants. As they decompose organic matter, microbes secrete organic compounds that bind soil particle together, improving soil structure and porosity. So, well-structured soil hold water better and give a root rooms to explore. So beyond this housekeeping roles, microbes act as a remediator. So in biocontrol, beneficial strain outcompete or antagonize pathogens and act as bodyguard for plant root. And in bioremediation, they degrade pollutants such as pesticide or heavy metal, helping to detoxify contaminated soil. Together, this function highlight how soil microbes act as nature's engineers, supporting both plant and soil health and environmental sustainability. Imagine assembling team of soil microbes, just 5 to 10 microbes per the trait that you want, and growing them together under controlled lab conditions. That's essential what a synthetic microbial community or SYMCOM is. So it is a simplified, tractable model of the complex microbial ecosystem in the soil. So the goal is to help microbes team up so the community stays strong and steady. So, one key detail that I have mentioned in the slide is the most commonly used microbes in synchrome constructions. So, the pie chart here shows that Bacillus, which contribute 41%, and Pseudomonas, which contribute 35%, dominate due to their well-known role in plant disease and depression and plant growth promotion. So the other frequently used genera include Pentoa, Enterobacter, and Streptomyces. So this is the study done by Martin et al. 2023. So these five genera combines robust lab handling, deep genetic insight, broad spectrum plant supporting activity, and mutual compatibility, which makes them go to building blocks for a synthetic community. So by engineering these syncoms, we not only uncover the fundamental rules of microbial cooperation, but also unlock the practical applications like outcompeting harmful pathogen to protect crops, producing organic acid and phytohormones that stimulate plant growth and developments, and breaking down pollutants in bioremediations, and ultimately reducing reliance on chemical fertilizer and pesticide. So, syncoms offer a powerful tool for harnessing and enhancing the natural functions of microbiome to tackle challenges in agriculture for environmental sustainability and beyond. So, talking about how syncoms are constructed, so it involves some complementary strategy that together turns a complex microbiome into a defined and high performing microbial squad. So, first, isolation culture. So, in this method, you start by collecting samples from soil or root zones and plating them in the selective media to generate hundred of individual microbial isolate. So each strain is then screened for trait you care about, say nitrogen fixtures or antifungal activity or any hormone production. And the top candidates are combined in a small consortia. So through iterative mix and match screening, as shown in the picture below, so you can just whittle down dozens of possible mixes to optimal multi-strain cocktail that stay stable in cultures. So for the second one, core microbiome mining, high throughput sequencing of the rhizosphere from healthy and thriving plants reveals which taxa recur across samples. So those core species are a strong contender for inclusion because their consistent presence suggests that they play key role in nutrient cycling, pathogen suppression, or plant signaling. So the automated design, so it includes the computer model ranging from genome scale metabolic reconstruction to the machine learning algorithm to predict which strain will cooperate best. So this in silico approach prunes the search space, pinpointing promising consortia to validate in the lab. So the last one, which is the most advanced one, gene editing based syncom construction method. So here you can fine tune individual member before assembly by deleting or overexpressing gene using different methods like CRISPR's, mutagenesis to boost specific functions like resistant gene for fighting with plant pathogens or enhance plant growth promoting genes. Syncom protect plant from disease through biocontrol strategy like competition for resource, antibiotics, needs colonization, induce systemic resistance, and by secreting volatile organic compounds. So for competition, by rapidly consuming root zone nutrients, sugar, amino acids, Syncom members are starve out potential pathogens. So think of it as a microbial musical chairs. If beneficial strain occupy every seat, invaders can't find food or space to establish. So antiviruses, many syncom bacteria secrete antibiotics, lytic enzymes, and high affinity siderophores. This molecule either directly kill or weaken pathogens or sequesters nutrients so effectively that the pathogen can't grow. So for fast forming biofilm on root surface, create a physical barriers so the the dense matrix of exopolysaccharide and cell blocks pathogen for attaching, penetrating, and accessing the root epidermis. For talking about induced systemic resistance, beneficial microbes display some microbe associated molecular pathogen that prime the plant immune system. When a true pathogen arrive, the plant mount faster and the stronger response, much like a vaccinated immune system. So the VOC, these are the gases such as acetone, which diffuse through the soil layer surface, inhibiting spore germination and hypergrowth of fungal pathogens. So I will be talking about two case studies. The one is done by In et al. For harnessing synthetic microbial communities syncoms to protect with seedling from soil-borne fungus Rhizoctonia solani. So they began with the Aploa wheat variety, which is highly susceptible to seedling blight. So from healthy wheat rhizosphere, they isolated a 14 bacterial strain So using those isolates, and they design 10 different syncums. So this community composition is based on following criteria, like different strain of the same genus, relatively strong antifungal activity in single strain tests, random combination, and combination of all 14. So that is mentioned in the table here. So they perform dual culture in lab and greenhouse experiment. So in dual culture, like 4 out of 10 symptoms reduces Rhizoctonia solani, showing the highest inhibition of 37%. And in greenhouse, 7 out of 10 symptoms significantly decreased seedling blight compared to untreated control, demonstrating robustness under like realistic conditions. So together, this finding illustrate multi-strain consortia can leverage complementary mode of action to deliver more consistent protection than the individual strains. So this is the second case study done by Feng and colleagues by asking the question like, can a synthetic consortium of beneficial rhizosphere bacteria protect potatoes in the field more effectively than single strain? So the pathogen is Phytophthora infestans, which cause the devastating disease called late blight. Maybe you all have heard about this. So, to answer their question, they isolated 8 candidate bacteria from the rhizosphere of the potato field, cultivating the Jizang 12 potato variety. So, they compare each strain and dual strain to an 8 strain syncom, combining all of them. So in their in vitro screening for the dual culture assay, the single strain B and 8 strain syncoms showed the strongest inhibition of Phytophthora, while like the 2 strain combination had moderate effect. So in the field experiment and the microbiome analysis, the 8 strain syncom reduced the disease and exhibited higher overall microbial diversity and altered community structures, suggested that the syncoms not only fight the pathogens but also reshape the soil microbiome in a more resilient uh, and disease suppressive direction. So the overall significance of syncoms is that they strip the soil microbiome down to just the strain you need. So instead of 1,000 species, you are working with 5, 10, or few dozen that you understand inside and out. That low complexity means interaction become predictable, letting you know who is doing what. Because you have defined every members, you also have high controllability. You can swap in or out a strain. Once you have got a stable mix, it stays consistent over time, which is essential if you are spraying the syncom products in field or coating seed. So basically farmer needs reliability season after season. So you can choose each microbes for its jobs, like outcompeting pathogens or promoting plant growth. So the community delivers targeted function on demand. So in short, like Syncom gives us precision, predictability, and performance in one package. So, looking ahead, the field is shifting from proof of concept community to truly designed, deployable product. First, researchers are working on rational design, assembling microbial consortia with predictable performance, scalable manufacturing, and self-stable formulation so that Syncom can be commercialized like any other agricultural product. Second, advance in genome editing and synthetic biology can help fine tune each syncum strains trait, boosting stress resilience or optimizing metabolite productions or adding environmental sensors so that the syncum adapt and thrive in the variable conditions of a real scenario. And finally, by discovering and modeling new microbiomes from soil and crop, we can expand our tool of beneficial microbes, creating farmer friendly mixer tailored to local climate, soil, and cropping system. Thank you all for your time and attention. I would like to express my sincere gratitude to my amazing Freedom Lab team members, especially Carol for this opportunity, and thanks to Pacific Northwest Farmer Network team to give me this opportunity to talk about Syncums. So if you want to know more about Syncum, please keep an eye on Wheatbeat podcast for their upcoming episode. Okay, thank you all for listening to me.